Cell Cycle and Regulation in the Cell by Alexis Porterfield. The cell cycle is an order of steps that take place for the cells to grow and divide into two new daughter cells. These steps the cells move through are specifically timed and cautiously monitored stages of growth in the cell. These two new daughter cells are exact copies of the former cell. Now, because this is called the cell cycle, we must understand that there is no beginning and end. Cells are continually entering and exiting the various steps in the cycle. So in the diagram, the outer strip shows the two main phases of the cell cycle process. We have the interphase and the meiotic phase. Notice that the cell spends more time in the interphase, and the interphase consists of three phases the cells goes through. You have a G1 cell phase, and then you have S phase and G2 phase. We'll go further into detail on the following slides. And then the meiotic phase this is the mitosis that we all might be a little bit more familiar with, and cytokinesis. This also will be discussed further in further slides. So the cell cycle, it's controlled by protein kinesis and that respond to cell signals, and these signals select the specific proteins needed to coordinate the activities needed to keep all this cell division in an ordered fashion in order to have those two new daughter cells formed at the end of the cycle to be able to be mature cells that will then be sitting in G1 phase. So cyclin and cyclin-dependent protein kinesis, these are the cyclin-dependent kinesis. They're a catalyst to help these to help regulate the cell division. They are very important in the phosphorylating of proteins on the amino acids, serine and threonine, that control what they do. So this diagram here gives you the different cyclin-dependent kineses and cyclins that are used throughout mitosis and the cell cycle. And over here, it's showing the phosphorylation. Okay, there you have the active, and when it's added to that phosphorus, then this one becomes inactive, or it can be the other way. You can have something inactive, and the phosphorylation makes it active. So there's about 10 different cyclins that are used, and 8 different cyclin-dependent protein kineses that work together in the several areas of the cell cycle. And they pair up in different orders to keep the cell division on track and following the necessary order for correct division, cell division. These signals made from the completion of each cyclin and cyclin-dependent protein kinesis interaction, it starts the next event to happen in the normal cell division, which is important in keeping those mutations and cancers from happening. It's also important to know there are checkpoints throughout the cell cycle that we'll talk about a little later. And this is where the cell must receive a specific signal to be able to continue on in the phase. And the signal can be the cyclins or protein kinesis. And these protein kinesis activate or deactivate by phosphorylation, which is the adding of that phosphate group between those molecules, which I was talking about over here. It was also important to understand that these cyclin-dependent kineses, they cannot do anything in the cell cycle by themselves. They have to be activated by those cyclins meeting up with them. And that's kind of what it's showing here in the diagram. You'll have that cyclin-dependent kinesis meeting up with that cyclin B, and together they can degrade meiotic cyclins. And down here it's showing you have the during the S phase, you have the cyclin-dependent kinesis 2 meeting up with the cyclin A, and those two come together and they do a function inside the cell cycle as well. Next is the interphase. So interphase was separated into 3 different phases, but we need to mention gap 0 so that we don't forget about it, because it does happen and there are cells that stay in gap 0. So interphase, of course, it takes up most of the cell's life, and before we got into the gap 1, I wanted to talk about gap 0. So gap 0, or G0, is where the cell began in G1, but it doesn't finish all the way through G1. It kind of gets stopped here at a checkpoint, and it doesn't make that checkpoint, so it continues to cycle in G1. It's important to mention that this occurs in some somatic cells that quit or leave the cell cycle, they called it. This is called the quiescent phase. It can be a temporary, resting, or quiet period for the cell, or the cell can be in senescent phase where the cell will never divide again. So neurons are something that was mentioned that never come out of gap 0. Liver cells and adipocytes, which are storages for fat in the connective tissues. These are a few particular somatic cells that enter the G0 phase. These cells that enter G0 phase are from a checkpoint in the G1 that can be caused with no growth proteins, or they could have gotten specialized functions and forms that keep them there. The cell cycle mechanisms are then taken apart in these cells, and the proteins, such as those cyclins and cyclin-dependent kineses, are not available anymore, and that cell stays in G0 until there's some reason for that cell to begin completing G1 again, and this means that the mechanisms of those proteins are returned to that cell, and then it can then continue on the cell cycle divide. So therefore it is important to know that that resting phase is reversible. Next is interphase of the gap 1 or G1. So looking under a microscope, the interphase appears to be very little activity going on. So at first it was believed that not much goes on during interphase. But they've figured out, scientists have seen that there's actually a lot going on the biochemical level during this phase. and the cell, it's beginning to grow in size because it's stocking up on proteins and RNA, and this phase makes sure that everything is ready in that cell for the DNA synthesis, and the cellular organelles are all being duplicated. So it's pretty much preparing the cell to move on, to be able to move on through the rest of the cell cycle, of course, and then enter that mitosis strong. So this phase, it takes up about 40% of the time in the cycle, and this means that when we need to think about what the nucleus and the DNA looks like before it goes through mitosis. So if you can picture, or the picture on the right, the left, it shows a nucleus, and you see all those stringy looking things, and those are all the chromatin. So this gives us a good idea of what it starts out as. And in order for it to be able to go through mitosis, that chromatin, it has to be condensed into chromosomes. And the chromosomes are those X-shaped, the X-shaped chromosomes that we're more familiar with. Now, with the help of proteins called those histones, the DNA can wrap around those histones, and then it folds into the X shape that we call the nucleosomes. And essentially, this is what I'm referring to. So you see all these stringy looking things. It's our chromatin inside the nucleus. And over here, the chromatin is then that string, and it starts to be wrapped around these histones. So these histones are really important. If we go back to thinking about the whole GAP1 phase, and what's going on there is it's stocking up all those proteins because it needs all these proteins. Histones are proteins. And if it didn't have enough histones, then it wouldn't be able to complete the folding of the chromatin into the nucleosome, which is what those X-shaped chromosomes are. So with the need of millions of those histones, this to tightly coil up those chromosomes, and chromatin enzymes, they're at work doing this phase, and they are facilitators to the restrictors of these gene expressions. So during the GAP1 phase, there's a checkpoint to make sure that all that DNA is in good shape with no damage, and the growth factors are accounted for, and all the necessary nutrients for that cell to complete G1, down here, G1 interphase, everything's got to be there. And there's that checkpoint that makes sure it's there. If it's not, then it won't be able to make it past G1 into the S phase. But if it is, it has everything it needs, it has all the histones it needs, the chromatin's ready, coiled up, then it can move on past that checkpoint in G1 into the S phase. So next is the S phase, and this is interphase S phase, the second part of interphase. And once these chromosomes are tightly coiled up, the S phase can begin. And during this stage in interphase, the DNA is being replicated into sister chromatids that are exact copies of each other. So these sister chromatids are connected by the centromere, and the centrosome is replicated for the centrioles that will be in charge of those meiotic spindles during meiosis, which then will pull apart the chromatids while they are separating for the DNA to go into those two new daughter cells. So to do the DNA replication, there are several important enzymes that work to make this happen. Helicase is an enzyme that separates or unzips the two strands of DNA. DNA polymerase is the enzyme that replicates and builds the new strand of DNA. Primase is the initializer, and this enzyme makes the primers out of RNA, which directs the DNA polymers where they need to be replicating and building those new DNAs. So eventually, the RNA primers are replaced by that DNA, and the DNA ligase, they fill in those gaps where the new DNA was made, so that they're kind of like the glue that seals those bonds. And then there's the topoisomerase is the enzyme that regulates those winding of the DNA. And during the S phase, there is another checkpoint for any damaged or not finished DNA replication. So in part of the S phase, there's the DNA replication. And I mentioned the different proteins that are used in DNA replication. So the DNA starts at the point of origin, and there can be more than one point of origin. At this point of origin, the topoisomerase puts the tension on that strand so that helicase can unwind the DNA and begin to separate that DNA, the double helix, by disrupting those hydrogen bonds and creating what is called a fork. So it's, if you think about it, it's the helicase is unzipping that DNA. Some single-stranded binding proteins, they come in and they keep those two strands that helicase separated, keep them separated so that primase can come in and make the RNA primers on each strand, which is necessary for that DNA polymerase to know where to start building those new strands that is the complement for the original DNA. So it's important to note that the leading strand is called that because the DNA polymerase can only build in the 5' to 3' direction. And everything runs pretty smooth. So this is the leading strand on this side because it runs from the 5' to the 3'. And compared to the lagging side, where it's going from the 3' to the 5'. And you can see these Okazaki fragments here. And this is where ligase enzyme comes in and it glues bases right in here to fill in that gap that would be there if the ligase was not there. And that you can see, or you can understand why the lagging side takes longer, because it still has to work on filling in those spaces from between those Okazaki fragments. So this all leads to those two new identical DNA strands that will be given to the new daughter cells once mitosis has went through. And then that brings us to the interphase of gap 2 or G2. And this is where the cell builds up its energy storages, and it there's a production of more proteins, all of which are needed for the moving of those chromosomes during the mitosis. There's also more organelles that are made, and the cytoskeleton is broken down so that it can be used in mitosis as a resource. And all of this causes that cell to grow more in size. So there's another checkpoint here towards the end of G2, right around here. And this checkpoint, it requires, or it makes sure that the cell is ready for division. And it's a B type. It's also known or called the mitotic cyclins, and they're made during that S and G2 phase, and they match up with the cyclin-dependent kinesis to make that maturation promoting factors that will keep the checkpoint or the factors that will let the checkpoint be passed if the cell is ready to move on to the mitotic phase. Now, if it's not ready to move on, then it won't be able to. It will stay into that G2 phase until it is ready to move on to cross that checkpoint into the mitotic phase. This is a good diagram that's showing those different checkpoints that I've been discussing. So the G1 checkpoint, as I mentioned, it's checking for the nutrients, making sure that there's nutrients for that cell to move on to the synthesis phase. Growth factors are going on, and there's no DNA damage. If there was DNA damage, then the cell would go into another phase that would end up killing the cell, and we'll talk about that in a little while. And then checkpoint 2, make sure that the cell size is good and the DNA replication is all good before it can move on to the meiotic phase. Excuse me. So then, once that cell is ready to pass on into the mitotic phase, then the steps in which the cells divide into those two new daughter cells containing those exact copies of DNA occurs. And the mitotic phase is separated into those two phases. There's kerokinesis, meaning that it's the division of the nucleus, and then there's cytokinesis, meaning that there's the separation of the cytoplasm and those organelles into those two new daughter cells. The cyclin used in the G2 phase gets degraded later on in the stages of mitosis, and the cyclin-dependent kinesis are inactive. And the steps in the mitosis make up the kerokinesis. Cyclin A and cyclin B couple to the cyclin-dependent kinesis 1 to make that cell that cell move through mitosis. And mitosis steps are all over here. So interphase, of course, the coiling of the chromatin, and then you have prophase. During prophase, the nuclear envelope is broken down, and those spindle fibers appear in the centromeres. These guys over here. And then as it moves through to metaphase, all those chromosomes line up down the center. Our spindle fibers are out, attaching to the chromosomes. And there is a checkpoint in the metaphase step where it's checking to make sure that those chromosomes are lined up correctly before that cell can move into anaphase. If they're not, then it will fix whatever it needs to do, and then it can move on to anaphase, where those spindle fibers start to pull those sister chromatids apart, and the spindle fibers start to break down as it's reeling in towards the separate poles. And then we have telophase, where the nuclear membranes begin to form around, and those meiotic spindle break down, and finally cytokinesis, we have the two daughter cells. I want to mention over here, this is a great diagram showing the different cyclins that are used during the cell cycle, and where they kind of so cyclin D, it's seen throughout the whole cycle, but it kind of reaches its peak at the middle to end of the S phase and starts to teeter down. And like I mentioned, cyclin B and cyclin A are used during mitosis. Cyclin A is seen a lot in the G2 phase, and cyclin E is kind of at that the transition from G1 phase into the S phase. And I just wanted to mention, when there's missed checkpoints, there's uncontrolled cell cycles, and those uncontrolled cell cycles will then have a higher possibility of turning into tumors and cancers, and that's why we have those checks and balances in the cell cycle, discussed in the previous slides. A couple of things that make that uncontrolled cell cycle happen are environmental factors such as carcinogens, pollutants, and radiation interacting with the DNA can cause single cell damage, and from those mutations that might occur. And lastly, a recap. So cyclins, cyclin-dependent kinases, and protein kinases are all hall monitors, if you will, of the cell cycle, and they get the cell cycle started in gap1 phase of interphase, making sure that there are enough histones for condensing and folding the chromatin into nucleosomes. If the cell is to stay in gap 0, then cyclins are gotten rid of and will not be brought back into the cell to work with those cyclin dependent kinases unless the cell for some reason needs to start the process to make it to mitosis. A checkpoint in G1 makes sure that there's no issues with the DNA and all growth factors are accounted for, and the nutrients needed is present. In the S phase, the protein enzymes are hard at work separating and building those new DNA strands to be given to those new daughter cells. In G2, it does more of the growing, checks and balances to help make sure there's protein kinesis for no problems with the DNA, and the cell is ready to move on to the mitotic phase. And finally, during that mitotic phase, we have those steps of mitosis, separating those sister chromatids and the formation of those two new daughter cells to make it through cytokinesis with all those extra organelles and the cytoplasm that they need to begin their own cell cycle in their own G1 phase. Thank you. Some resources.